Hello everyone, and welcome to Biopedia. Today we're going to zoom in to cover the enzymes known as GTPases and the role they play in cell polarity and movement. This necessarily means that we will have to cover a lot of topics that we will essentially be skipping over, but when and if I release appropriate episodes on these, I will link them in the description. So before we dive in, we need to talk about what cell polarity actually is. Polarity is simply the act of having one side that is different or distinguishable from another along an axis. For example, humans have a front and a back, a left and a right, a top and a bottom. These two sides are distinct from each other, so we are polar organisms. All organisms are polar to some extent. For example, if a bacterium has a flagellum, then there will necessarily be one side where the flagellum is present and one where it is not. This polarity, however, needs to be set up at some point within the cell as it develops, and, although it is a big topic that we will likely come back to in future episodes, I am here specifically going to focus on the fact that cells need to change their shape and their polarity when they react to their environment, such as when chasing or finding food. Specifically, I'm going to focus on the enzymes known as GTPases in this context. A GTPase is an enzyme which can cleave guanosine triphosphate, or GTP, into guanosine diphosphate, GDP, by removing a phosphate group. However, it is itself regulated by which version of this same molecule is bound to it. When the enzyme is bound to GDP, it is inactive, while it is active when bound to GTP and can thus act on other molecules. Other molecules can thus consequently regulate the enzyme's activity. An enzyme abbreviated as GEF or GEF activates GTPases by swapping the GDP bound to the enzyme with the GTP, essentially recharging it. By contrast, an enzyme abbreviated as GAP does the opposite and inactivates it. GTPases are crucial because they have many different substrates that they can act upon, which thereby means they can regulate the activity of many different proteins. If these target molecules are themselves enzymes, such as kinases, this can cause a cascade of reactions, such as the kind that regulate cell polarity and shape. For cells to be able to undergo this shift in polarity, the cytoskeleton plays a crucial role. As with cell polarity, we're going to come back to the cytoskeleton because covering all these topics at once would be far too much information to deal with. Again, when I release a relevant explanation episode, I will link the two in the description. For now, we're going to focus on the actin cytoskeleton and its interactions with GTPases. In mammalian cells, there are three different kinds of GTPases, Rho, RAC, and CDC42 families, which interact with different parts of the actin cytoskeleton. Rho GTPases interact with adhesion sites, which are the points where the cell comes into contact with the extracellular matrix. RAC GTPases cause changes in a structure called the lamellipodium, which is a broad sheet of actin filaments at the edge of a cell that enable a cell to migrate by generating pressure on the cell membrane, a mechanism which again we will come back to. Finally, CDC42 ultimately interacts with actin in filopodia, which are long projections out from the main body of the cell which require long parallel bundles of actin. As the actin cytoskeleton is crucial for cell movement, enzymes which ultimately influence this cytoskeleton have a big impact on polarity and thus movement-associated changes to the cell's orientation and polarity. GTPase activity can also be influenced by outside stimuli rather than purely intracellular ones. For example, a ligand can bind to a particular kind of receptor in the plasma membrane known as G-protein coupled receptor, or GPCR for short. This name refers to the fact that the binding of this ligand to the receptor triggers a conformation change in a 3-molecule GTPase complex bound to the plasma membrane. A series of intermediate steps then lead to the activity regulation of GTPases themselves. In brief, this ability to connect the regulation of the actin cytoskeleton, as described above, with external stimuli, in the form of, for example, the G-protein coupled receptor, allows cells to move in response to their environment, which is obviously an important process. This episode, by necessity, required the mention of a few terms that we have not covered yet, so I will ensure that when we get to these episodes, I will bring this one up again in order to connect the concepts more thoroughly. In the meantime, I hope you enjoyed the show. We are available as always at biopediapodcast@gmail.com for any comments, topic requests, or questions. Thank you for listening, and until next time.